Welcome to MVIF Conversations, the podcast.
Hello everyone, and welcome to the first episode of MVIF Conversations, the podcast. My name is Stefanie Malan-Müller from Complutense University in Madrid, Spain, and today I'm thrilled to be joined by Professor Jack Gilbert from the University of California, San Diego. Professor Gilbert is one of the leading figures in the world of microbiome research, and his work spans from the microbes living inside of us, shaping our health and impacting disease, to those that inhabit our oceans, soils, and even our buildings. His curiosity truly stretches across the full spectrum of life on Earth and beyond. I'm joined today by Christina Pavloudi from the European Marine Biological Resource Centre, whose focus lies in the environmental side of microbiome science, while my own research interests lie in the human health side. So together we'll be picking Professor Gilbert's brain about the fascinating intersections between humans, the environment, and the microbial world that connects us all.
Jack, welcome to the show. It's really great to have you here.
Great to be here. Thank you so much for the invitation.
To start us off, could you share what initially drew you to study microbiomes across such a broad range of systems, so from people to the planet? What's the common thread that keeps you curious along all these worlds?
Probably ADHD. I don't know. I just like shiny things. And when the next awesome story comes up, I get really excited by it. I love stories. And so I've always been kind of drawn to trying to understand how things are connected and trying to tell stories and trying to understand how those stories interact with different things. So for me, I think the first time I saw that was when I was doing my PhD. I found this particular bacteria and it does this really cool stuff in an Antarctic lake. I just thought that was awesome. And then, there's bacteria everywhere. So I could find those cool stories in every ecosystem on the planet, including our bodies. And so that really drove me to try and understand how those stories function in different worlds.
It's really an interesting story. So you've published extensively on the human microbiome, from early foundational studies to new work in precision health. Where do we stand today in terms of moving microbiome science from bench to bedside? Are we getting closer to seeing microbiome-based diagnostics or treatments routinely being used in clinics or hospitals? And are there particular areas like gut health, oncology, or metabolic diseases where you see most promise for near-term applications?
Yeah, so there's obviously three therapeutics nearly that are approved already by the Food and Drug Administration in the United States. And there are a number that are essentially approved in Europe and Australasia and in China. And those are therapeutics that are mostly focused around the treatment of infectious disease in the gut. And so that, we have already made that an initial transition to a translational microbial technology. Diagnostics is something different. There aren't currently any that are effectively delineated and approved, but that to me is the biggest possible benefit of microbiology in human health, is being able to use this extremely highly dimensional and extremely dynamic ecosystem to augment our ability to predict who responds to a therapy, to predict or diagnose a disease in an individual, to look at risk for therapeutic outcome or for disease transition. We just had a paper a couple of years ago where we looked at the oral bacterial community and its direct association being the important word, no mechanism or causality defined, but it's direct association with the progression of idiopathic pulmonary fibrosis. This is a chronic fibrotic disease of the lung, which damages the lung beyond repair and invariably leads to death. We can now predict, based on oral bacteria with very high degree of certainty, the state of that disease and the progression rate of that disease in an individual. And we have loads of examples, like in cancer, we found that bacterial enzymatic activity at the point of, say, colorectal cancer surgery to remove the tumor, the bacterial enzymatic activity in your stool can be extremely predictive of your risk of a recurrence of your cancer. And we can go into the mechanism and try and understand all that. we also know that microbes can help us to predict and also be used as a therapeutic for, say, the transition or effectiveness of an immunotherapy for cancer. And this stuff you can't help me. It's like picking my favorite child, right? I'm not going to Sophie's choice the different strategies that microbiology can be used in clinical therapeutics. There's just so many of them, and they're so vast that it really is an incredibly exciting field.
Truly. When we talk about the gut microbiome, most of the conversation still revolves around bacteria, but of course that's only part of the story. There's also fungi, archaea, and an incredible diversity of viruses and phages. So how do you think we can move towards a more holistic understanding of this complex ecosystem, and what needs to happen technically or conceptually to capture the complexity, both in research and in clinical practice?
Yeah, I mean, I think for the clinic, it's incredibly difficult, right? There's some brilliant studies that have demonstrated that if you take a germ-free mouse and you colonize it with just virus particles, that's phages and eukaryotic viruses, you get the same kind of immunogenic activity as if you colonize it with just bacteria or just bacteria and archaea. So there's an important premise there, right? That the viruses have the same level of antigenic activity in the body, potentially, especially for mice, as bacteria and archaea do. So what's the difference? What role do they play? That's reason science is here, right? We have to tease apart the mechanistic relationships there, try and understand the toll-like receptors which are activated for each component, and try and understand how the human body or animal's body interacts with this diverse microbial ecosystem, both metabolically and immunogenically, and then try and really tease apart which pieces are vitally important. But to me, that's the easiest step. One of the most complicated steps is trying to create models, mathematical and computational, say, digital twins of that ecosystem, and tease it apart at an individual level so that we can take specific input metrics and predict how that microbial ecosystem will respond to an individual's healthcare journey or an individual's treatment strategy. To me, that's much harder and much more exciting than just, say, trying to figure out which strains of certain organisms might be valuable. We already know, say, probiotics can be valuable. I think there should be probiotic viruses. Right, we've been using them for a while in immunology in order to immunize people against diseases. We could use them effectively to drive forward healthcare functions as we do with probiotics. There's nothing wrong with that strategy. It's just not been implemented, mostly because of social fear. But we can overcome those if we can package them effectively and work with industry to figure out ways of communicating that to the public. But I think creating those digital twins of how viruses, bacteria, archaea, fungi, protists interact in an ecosystem and interact with that particular unique ecosystem that they find themselves in, say in the colon or in the duodenum or in the stomach or in the alimentary canal or on the skin or in the vagina or in the in the lung, is incredibly important. It's just it's not a small, easy thing to do. It's going to take a while for us to parameterize those models.
Yeah. Thank you. Thank you very much for this answer. Just to take you on the more environmental side of your work, just some months ago, the International Union for the Conservation of Nature, IUCN, officially approved the creation of the Microbial Conservation Specialist Group, the first ever IUCN Species Survival Commission group dedicated entirely to the protection and inclusion of microbial biodiversity, which you are, of course, co-chairing with Raquel Peixoto. You have also co-authored the call to action demanding immediate, tangible steps that harness the power of microbiology and the expertise of researchers and policymakers to safeguard our planet for future generations. One of your actions would be to publish a global microbial hotspot map. Well, global microbial hotspot maps in plural. How difficult do you think this task would be, given that there are some major knowledge gaps, especially in the global south? And are we risking favoring certain ecosystems such as hot springs or coral reefs that are, the usual suspects of research focus, while neglecting other ecosystems?
Yes. That's the biggest question I think I could possibly ever face to answer. There's about 50 parts to that question, and I don't know how to break it down effectively. I just got off a call prior to this meeting with the Red List community for the IUCN. So one of the commissions associated with trying to identify the extinction risk and or restoration potential of particular ecosystems or particular species, right? And just trying to figure out how to shoehorn bacteria, for example, into that paradigm is difficult. I would say we've created an immense diversity of people to tackle the diversity of ecosystems. So I'm less worried about us not being able to cover ecosystems around the world, including in the global south. We have big data gaps and knowledge gaps about biodiversity on a global scale in virtually every part of the world, including in America. And you wouldn't have assumed that, right? But there's huge areas of America which we have no knowledge of whatsoever, microbiologically speaking. So I think that's a universal problem. But I do think it's incredibly important that we delve in as a global community to try to understand how to integrate microbiology into the conservation strategy. And let me just tease that piece apart, because I think it's important, right? The first one is I'll start with the second one, actually, because that's more exciting. So the second one is we can use microbiology to enhance conservation. What does that mean? Well, that means using it to enhance the fecundity of otters and rhinos and the preservation of sequoia trees and whatever it is you want to preserve, right? Pandas to sloths. We can use microbiology to enhance their health, to enhance their nutritional impact, to support their restoration and their population growth. We know this. We talked about this in human health. We can do the same thing in animal health and plant health, and that's the really important piece of how microbiology can be used to effectively support conservation more broadly. We can also reintroduce, if we know what's at threat, we can capture the microbiology in seed banks and culture labs and culture collections, and we're working with many of them around the world right now. We can do that, and then we can reintroduce that microbiology into an ecosystem to support the restoration of that environment, but also to support the restoration of those bacterial species which are under threat in a given ecosystem. The hardest piece about this is figuring out what species of bacteria are under threat. So, okay, let me give you the best example which I can come up with, and this is an age-old example, but it's pretty useful for getting human brains engaged in this story, right? Bifidobacterium longum infantis. Coming back to human health, humans are just part of the ecosystem, so I think it's fair, right? But coming back to this one particular species, it's a uniquely adapted species, subspecies, one might say, of Bifidobacterium longum that's uniquely adapted to breaking down human breast milk sugars. And it has nearly the entire genetic enzymatic repertoire of capabilities for breaking down the vast array and fluctuating over the lactation life cycle of a woman and a baby, breaking down all those sugars across that trajectory. And it's very, very good at doing it. It's also disappearing from developing societies like China and North America and Europe and certain parts of sub-Saharan Africa, Australasia, etc. These communities are seeing the abundance and presence, more likely presence, of this species disappearing, right? Mm-hmm. This is a given. What do we do about that? Well, you could say we need to protect and conserve the genetic diversity of Bifidobacterium longum infantis, because the strains of that species, that subspecies that are present in, say, populations in Ghana are going to be very different from the strains that are in populations in, say, Argentina, right? And understanding you can't just go into Ghanaian populations, culture up the bacteria, grow it and throw it back into populations in Argentina. We don't know that will work. We don't think that's effective. And also that will lead to a global monoculture of Bifidobacterium longum infantis, which, as we know from plants and animals, is not a good thing, right? So we need to protect and conserve the genomic diversity of this species and leverage that diversity to enhance human health and resilience more broadly at a global scale. So, when you dig in, and I got examples of that from prairies and mangrove swamps and coral reefs and panda bears and whatever you want to throw at me, I'll find a great example. But global oceans. But it's really important that we figure out as a community how to integrate microbiology and how to fit the current conservation programs around these microbiological questions. How do you red list a species like Bifidobacterium longiminfantis? It doesn't have a generational gap. It doesn't have a generational age, really. I mean, it breeds once every 40 minutes or so. So that's a bit different to a panda bear, which breeds about once every 2 or 3 years. And when you're looking at red lists, you often understand the number of mature individuals. Why do you do that? Because mature individuals replicate, right? What does that mean for microbes? They mature after 30 minutes or so. So what do we mean by mature? That doesn't make any sense. So we need to understand how to integrate current conservation paradigms into a microbial world, because this planet is a microbial world. Right? It's not a world for you and me and the plants and animals. That's ridiculous. It's a microbial planet, and these things have just come along in the last 500 million years or so. They're a relatively new introduction. So we need to better understand this planet from that microbial perspective, but we also need to work with the social creatures that humans are and fit microbiology into that existing paradigm. And that is a hard problem to do. And so hence we need the global community to come with us on this journey and understand how to integrate it. And we're all super busy. I totally get that. But I think we can do it and we can have some fun.
I also think we can do it. And I think it's imperative, and it's actually probably it was a need, a need come through this, this group and getting microbes into IUCN. So yeah, definitely, I think you will find all the collaboration and we will find many interested people to join you in this effort.
How do you say that? You say that, but that's where it gets really interesting, right? We're already getting some pushback, weirdly, from microbiologists. I've had microbiologists who have said to us and are starting to write papers saying, ostensibly, why do you need this? Why do you need this? You can serve an ecosystem, you can serve the microbiology, surely. If we can serve a coral reef, then you're conserving the microbiology as well. I think that's the wrong way to look at the problem. And I think therefore we need better, talking about stories, like we talked about at the beginning, we need better stories to help to bring along not just the conservation biologists, who all have expressed quite a lot of excitement about this idea, but also the microbiologists. And you and I know, as does everybody potentially listening to this, that you get up at a microbiology conference and you tell your story, you'll reach maybe 5% of the audience. There'll be another 35%, 40% of the audience which are really interested, right? And then the other 50% will be like, doesn't really apply to me, and I don't really understand it. And that's a microbiology conference with microbiologists, right? But we're such a diverse group of people that bringing us all along on this journey needs a really strong story that everybody can feel like they have a agency in, that they have the opportunity to become involved and that they see themselves as part of. So this is far more a sociology experiment than it is a microbiology experiment, and I think that's an important piece about bringing together transdisciplinary communities to help us to reach these goals that we have. And those goals are super large, right? These are magnificent goals, one might say. But they're also incredibly important, because if we don't do them, in 50 years' time, we're going to regret it horribly, and it's going to come back and kick us in the proverbial.
Yeah, I couldn't agree more. I think I will pass it on to Stefanie for the next batch of questions.
So one of the things I find really inspiring, and it connects well with the previous discussion, is your work and how it bridges human health with that of the environment. and the idea that the microbiomes of humans, animals, and the environment are deeply interconnected. So in light of that, how do you see the One Health approach evolving for microbiome science? If you can imagine an ideal world scenario where human, animal, and environmental microbiome data were integrated, what could that look like, and what kind of insights or benefits do you think it could bring?
Yeah, I think I've been answering these questions for 20 years or so, and I don't know if I think it has changed a bit, but I'd say my answer is still nuanced, right? One Health has been around for a very long time, and it's a wonderful concept that we're all interacting, and our bodies and the bodies of animals and plants are all part of one larger ecosystem, and we need to understand how the whole ecosystem health functions in order to support our health and the health of the world around us. And that to me is almost axiomatic. It's obvious, right? It's not a we're not isolated beings living inside our little beehives, and there's no interaction with the outside world. I'm right now breathing in mostly my own bacteria in this space, but there's also bacteria from my dogs and my children and my wife, and and the plants outside my window, because I live in a suburban environment with lots of plant diversity. All of that is shaping my health. And then when I get in my car and I drive to work, I'm exposed to different things, and all of that is shaping my health, and it shapes the health of those environments around us. We are connected. We're each emitting 30 to 40 million bacterial cells an hour from our bodies, mostly through our respiratory tract, but also from our skin. So we are shedding microbes into the environment, and by proxy, the environment's shedding microbes into us, right? And so that is a constant flow. If you will, the microbes are the immunological and metabolic language by which we communicate with the world. And so to me, it's rather obvious that if we don't shape the environment properly, our health is not going to be shaped properly. And a great example is work we did even like 10 years ago with Carol Ober and Donato Pacelli and Anne Sperling at University of Chicago in Arizona, where we looked at, say, children that are growing up in a very rural farming-based environment, such as the Amish in Indiana, versus a similar community that live a very similar lifestyle, but whose children aren't allowed to grow up in a farming environment, the Hudderites from North Dakota. And these communities, they eat the same kind of food, they have the same genetic history, the same risk of developing allergic disease genetically, right? But they have very, very different levels of disease. So the Amish have virtually 4% of their population develop asthma, whereas in the Hutterite population, it can be up to 25 to 30% of their population develops asthma. And what we found is that if you take the dust, which contains a lot of antigens, but the dust from the children's bedrooms, you sneak in like the Grinch into these kids' bedrooms and you suck up the dust from the Amish kids' bedrooms and the Hutterites' kids' bedrooms. That dust has different immunogenic properties. So we take that and you put it into an animal model. The animal model will have very different reactivity. In fact, we created an animal model that has a food sensitivity, sensitive immunologically to the protein of albumin, right? And if you give this animal of albumin, it will have an allergic reaction. So, okay, great. If we take the Amish dust and we sensitize this animal with Amish dust up its nose and into its lung and let it breathe it in, right? It no longer has an inflammatory response to albumin. But if you take the Hutterite dust, it still does. So there's something missing in the Hutterite dust that's present in the Amish dust, which helps to desensitize that animal to its allergic reactivity. These are really important pieces, right? How do you tease apart that? Well, how do you use that information to change human health? Well, you got to figure out how to get these kinds of immunogenic opportunities back into people's lives. And that's where probiotics and prebiotics and nutrition and brilliant work from Amber Fife Johnson up in Washington, which is taking children and getting them out into forest schools so that their bodies can experience the immunology of those forest environments and improve their health. These are all pieces where we have to figure out sociology, cultural dynamics, socioeconomics, government processes, and how our bodies interface with that system. And so, we work a lot with Sue Ishak from University of Maine, who developed the the Microbiomes and Social Equity Program, which is currently administered by the Applied Microbiology International Microbiology Society. And that group is trying to ask these really important questions about how sociology and government practices interact with our bodies through microbiology. And so this plays a part in our One Health story. How do we integrate One Health into our environment? Well, we do it by supporting our governments to make better choices about how they interact with our bodies through microbiology.
Fascinating. We really need to get everybody on board, everybody together, and to make this work. Perfect. Thanks.
We're getting close to the end, but we have a few more questions for our listeners. You have co-founded the Earth Microbiome Project in 2010, which aimed to construct a global catalogue of the uncultured microbial diversity of our planet, and the American Cat Project in 2012, aim, of course, to discover more about the microbiomes in our own bodies. What do you think is the next project? Would it be a second round of Earth Microbiome Project, a more regular monitoring scheme? Would it be a different kind of project?
Yeah, my next big exciting project that I'm really excited about is the opportunity to develop and build a accelerator, a global moonshot or a prize for microbiology, if you will, where we accelerate the translation of microbiology at scale for both industrial practices and commercial practices that are invested in supporting microbiological adaptation to technologies getting out there in the world that can support, global climate change and reduce the impact of global climate change, biodiversity loss, ecosystem restoration, food security, all of the UN Sustainable Development Goals, but at scale. So we are now pushing forward for the development of a global microbiome accelerator, where we will have a fund supported by the brain trust, if you will, of the global microbiome societies community. That's you, me, everybody that's involved in a microbiological academic society, to identify new technologies that are out there that involve microbiology and to seed them with non-dilutive capital to support their success in a industrial commercial market. So a great example of this is trying to identify companies that are explicitly looking at ways to improve soil health and improve agricultural food security for our populations. We need to wean ourselves off of nitrogenous fertilizers. They pollute the environment and they're incredibly polluting for producing climate active gases in their production and distribution. So we need to wean ourselves off of these products, right? We need to support active engagement of our agricultural communities in microbiology so that microbiological solutions can support their economic bottom line, make them more profit with less input, and also support the resiliency and stability of our soils to changes in climate. We can only do that by supporting agricultural companies in doing this process, right? So we want to say, hey, look, we're pretty good at microbiological science. Can we use that knowledge and information to identify which companies are coming up through the ranks, and can we then fuel those companies with the right kind of funding strategies and support mentoring networks to enable them to be successful? That to me is the best thing we can do. I can only go so far with federal, state, government funding and philanthropy. I need to move this into the commercial and industrial sector, where we have vast financial resources that will allow us to have a much, much bigger impact upon translating microbiology at scale. And that, I believe, is the only way we can tackle the global climate threat and the global biodiversity threat, is by supporting microbiology in industry.
It's a really great initiative. Before we wrap up, I wanted to ask a forward-looking question. Where do you see the field of human microbiome research heading over the next 5 to 10 years? And what do you think are the key strategies or workflows we need to implement to move closer to a broader clinical public health application?
The key thing, I could take that question in about 50 different directions. I'm not going to. I'm going to focus on something I'm interested in. the key thing I think is most important is that we can support human health at home and in the community. And we can do that through our clinics, through our doctors, through our nurses, through our health professionals. We can support people having agency over their own health. And the best way to do that is to support them making the right kind of lifestyle decisions. That's, mild exercise, it's getting out into nature, it's like maybe having a furry pet when they're babies, but it's also about nutrition. And nutrition is key. I run a precision nutrition initiative for the NIH. And these initiatives are focused on trying to identify how our bodies individually respond to different types of food, right? Why the three of us might have a very different response to pineapple, for example, physiologically, immunologically, metabolically. A lot of that appears to be mediated by the different types of bacteria which live inside our body. And then the impact is felt based on our different sex hormone levels and metabolic state, our birth sex and our ecosystem that we live in, physiological stress, psychological stress that is associated with our socio-political environment. All of these features play a role, absolutely, but we can more effectively support the use of, say, food as medicine in helping individuals to make more effective choices. We can't do that at a global scale because of food deserts, food swamps, and the socioeconomic differentiation of food activation in different populations. But we can make more effective decisions about how to provide people with effective food choices that are tailored to their needs and their socio-political environment, so that they can support their own healthcare journey in moving forward in our developed society. I don't think I can stop all inequity in our world, but I think I can make our clinical sector provide better and more effective food and nutrition advice to our patient populations, so that they can support their own healthcare journey more effectively. And at the baseline of that is microbiology, because food interacts with the microbiome, which supports our human health. So this to me is the biggest translational potential. I think if you fix the food problem, wean us off of our ultra-processed foods and get us more into a whole food system that's more equitably available, and make sure that your local grocery store is not a 7-Eleven, right? If we can wean ourselves off of that problem, then a lot of our chronic health diseases will also start to abrogate over the next 50 years. As we'll have a healthier, more effective population, maybe even a more fecund population, that will support our success and resiliency in the face of the upcoming threats from climate change and global instability. So to me, these are inexorably linked features that need to be embedded in our global healthcare system.
For sure. Thank you.
Thank you very much for this great talk and for sharing your insights with us today. It was a great discussion, and I really hope our listeners will enjoy it as well. So we've reached the end of our conversation. Thank you for tuning in to MVIF Conversations, the podcast. Remember to sign up for our free monthly virtual conversations. And until next time, stay curious, stay connected, and don't forget that microbes are everywhere. Thank you very much.
Thank you.
Thank you for listening to MVIF Conversations, the podcast. Until next time.